Let us now discuss the replication of DNA molecules. DNA molecules are the carriers of genetic information within a cell. That is, they are the molecules of heredity. Each time a cell divides, an exact copy of the DNA of the parent cell is needed for the new daughter cell. The process by which new DNA molecules are generated is DNA replication. DNA replication is the biochemical process by which DNA molecules produce exact duplicates of themselves. In a cell division involving mitosis, it produces two daughter cells that are genetically identical to each other and genetically identical to the parent cell. Remember that for this to happen, DNA is the parent cell must be replicated before the cell divides. This process occurs during interphase in the cell cycle. To understand DNA replication, we must regard the two strands of DNA double helix as a pair of templates or patterns. During replication, the strands separates. Each can then act as a template for the synthesis of a new complementary strand. The result is two daughter DNA molecules with base sequences identical to those of the parent double helix. Under the influence of the enzyme DNA helicase, the DNA double helix unwinds, and the hydrogen bonds between complementary bases are broken. This unwinding process is somewhat like opening a zipper. After the unwinding process, DNA molecule separates into complementary halves. The bases of the separated strands are no longer connected by hydrogen bonds. They can pair with free individual nucleotides present in the cell's nucleus. The base pairing always involves C pairing with G and A pairing with T. The pairing process occurs in one nucleotide at a time. After a free nucleotide has formed hydrogen bonds with the base of the old strand or the template, the enzyme DNA polymerase verifies that the base pairing is correct and then catalyzes the formation of a new phosphodiester linkage between the nucleotide and the growing strand. The DNA polymerase then slides down the strand to the next unpaired base of the template, and the same process is repeated. Though simple in principle, the DNA replication process has many intricacies. The enzyme DNA polymerase can operate on a forming DNA daughter strand only in the 5' to 3' direction. Because the two strands of a parent DNA run in opposite direction, only one strand can grow continuously in the 5' to 3' direction. The other strand must be formed in short segments. These are called Okazaki fragments, after the discoverer Reiji Okazaki, as a DNA unwinds. The breaks or gaps in this daughter strand are called nicks. To complete the formation of this strand, the Okazaki fragments are connected by the action of DNA ligase. Take note that the process of DNA unwinding does not have to begin at the end of the DNA. It may occur at any location within the molecule. This is a diagram example of DNA replication. The old strands will serve as a template which will be unzipped by the enzyme DNA helicase. New bases will be added. The pairing will be verified by the enzyme DNA polymerase. As mentioned earlier, the unwinding process can occur at any location within the DNA molecule. The enzyme topoisomerase unwinds the DNA, and the enzyme helicase is the enzyme that breaks the hydrogen bonds. The enzyme DNA polymerase is the enzyme that catalyzes connection of nucleotides to form complementary DNA strand in 5' to 3' direction. The DNA daughter strand 5' to 3' direction is called as the leading strand. The leading strand is a continuously growing strand, while the lagging strand is a strand growing in segments, or what we call the Okazaki fragments. The enzyme DNA primase is the enzyme that catalyzes the formation of RNA starting segment, or the RNA primer. And lastly, DNA ligase is the enzyme that catalyzes connection of two Okazaki fragments. Transcription is the process by which DNA directs the synthesis of hnRNA or mRNA molecules that carry the coded information needed for protein synthesis. Before we discuss the RNA synthesis, let us discuss first the different types of RNA molecules. RNA molecules found in human cells are categorized into 5 major types, distinguished by their function. These 5 RNA types are heterogeneous nuclear RNA or hnRNA, messenger RNA or mRNA, small nuclear RNA or snRNA, ribosomal RNA or rRNA, and lastly, transfer RNA or tRNA. Heterogeneous nuclear RNA is the RNA formed directly by DNA transcription. Post-transcription processing converts the heterogeneous nuclear RNA to messenger RNA, which carries instructions for protein synthesis to the sites for the protein synthesis. The conversion of heterogeneous nuclear RNA to messenger RNA is facilitated by the small nuclear RNA. The ribosomal RNA is the RNA that combines with specific proteins to form ribosomes, the physical sites for protein synthesis. And lastly, the transfer RNA is the RNA that delivers amino acids to the site for protein synthesis. The mechanics of transcription are in many ways similar to those of DNA replication. There are 4 steps involved. The first one, a portion of the DNA double helix unwinds, exposing some base. The unwinding process is governed by the enzyme RNA polymerase rather than by the DNA helicase. Free ribonucleotides, one nucleotide at a time, align along one of the exposed strands of DNA bases, the template strand, forming new base pairs. In this process, uracil rather than thymine aligns with adenine in the base pairing process. Because ribonucleotides rather than deoxyribonucleotides are involved in the base pairing, ribose rather than deoxyribose becomes incorporated into the new nucleic acid backbone. Aside from unwinding the DNA double helix, the enzyme RNA polymerase is also involved in the linkage of the ribonucleotides, one by one, to the growing hnRNA molecule. Transcription ends when the RNA polymerase enzyme encounters a sequence of bases that is read as a stop signal. the newly formed hnRNA molecule and the RNA polymerase enzyme are released, and the DNA then rewinds to reform the original double helix. A mutation is an error in base sequence in a gene that is reproduced during DNA replication. Such errors alter the genetic information that is passed on during transcription. The altered information can cause changes in amino acid sequence during protein synthesis. Sometimes, such changes have a profound effect on the organism. The substance or agent that causes a change in the structure of a gene is known as the mutagen. Two important types of mutagens are radiation and chemical agents. Radiation, in the form of UV light, X-rays, radioactivity, and cosmic rays, has the potential to be a mutagenic. UV light from the sun is the radiation that causes sunburn and can induce changes in the DNA of the skin cells. Sustained exposure to UV light can lead to serious problems such as skin cancer. Chemicals can also have mutagenic effects. Nitrous acid is a mutagen that causes deamination of the heterocyclic nitrogen bases. For example, nitrous acid can convert cytosine to uracil. The deamination of a cytosine that was part of the mRNA codon would change the codon.